Okay, sorry to leave everyone kind of hanging for the last couple of days. Hopefully, you've been able to watch those other lectures. I may be posting more of those. They tend to be much shorter, less long-winded than me, and actually very good. So, the thing that I can do is direct your studies. And so, what I've been trying to do is make sure the slides are put together well. And I've just come to realize a lot of this is a lot more complex than it needs to be. So I'm going to go through this little bit that is represented by those other lectures and just kind of focus on what you need to know. And so we're going to start in section 24.1C, the supercoiling of circular duplex DNA. And so within a cell, DNA is condensed, and we'll talk about this a little bit later, but if you took the DNA out of a human cell, it would be about 2 meters long. And so cells are actually fairly small, about 100 micrometers. And so taking this big, long molecule, not very wide, but very long, to pack it, you need to do some tricks. And so what you can do is what's referred to as supercoiling. So what we've got here is a circular form of DNA in the panel down here to the left. And you can see that it actually twists up. And so you can imagine that you just have a necklace and you start twisting that necklace. And so as you start twisting that necklace more and more, or even just imagine a rubber band that you just start to twist, that shape will start to condense and collapse into a smaller piece. Now, they have this formula here. Gosh, I spent a lot of time trying to figure it out. I don't like the images in the book, but what they have is this linking number and the turns and the ride number. Now, the key to this is that if we're going to join a piece of DNA into a circle, it already has some links. Some DNA is already forming a coil. So what we're actually doing is making a coil of coils. And you can kind of see the advantage to that in this diagram that would go from a linear molecule to a coil. And then if we coil that coil, it's actually going to be more compact. Now what we have is this linking number that essentially is the number of times that the DNA crosses over itself. Now, if we were actually going to make it into a closed loop, we would have to essentially remove one of those links and connect them. And so what it's showing here is that we link those two ends together. And so if you went through and counted each one of these points here, we would just have 9. And that is going to be the same number of turns in here. So this W, this writhe, is what happens when you start to supercoil. And what made sense to me is if you writhe in pain, you're kind of flopping it around on the ground and you're coiled up. Or the other thing that makes sense is if you take a rubber band and you start twisting it, so imagine this in your head, or grab a rubber band and start twisting it in the opposite directions, you're going to start to get some turns there. And pretty soon, those turns, as you add more and more turns, is going to cause that rubber band to kind of want to pull on itself or writhe. So it's going to actually kind of turn, and what you're going to see is that it's going to pull those ends in and start wrapping around itself. And so when you do that, you've increased the number of turns, and you've also added some writhe to it. Now notice in this bottom right example here, we still got the same number of linkage. That's going to be a constant number here. And we've increased the number of turns there, but to keep the linkage number the same, we actually have to add this writhe term. Okay, now I'm not too interested in this math. What you need to understand though is that this DNA, when it's circular, it's actually going to roll up into these supercoils. So essentially you've got these helices that are now coiled, and then when you start to add some more twist to these helices, they're going to coil the coils. Okay, and the advantage for that is you get more compactness to your DNA. So typical DNA in the B form has about 10.5 base pairs per turn. Okay, so naturally occurring DNA circles are underwound. So it leads to this negative supercoiling. And in order to process this DNA, what you need to do is actually nick it so that it will have a rise of 0 so that you can actually get in there and process it. Now, overwound beta DNA, so what that means is you are winding it in the opposite direction to the helix. And so that's going to have what's referred to as positive supercoiling. Underwound DNA, what's going to happen is you're in the same direction as the helix. And so as you underwind it, you're going to make it so that it has less than 10.5 base pairs per turn. And when you have less or more than 10.5 base pairs per turn, the DNA, you start to strain some of those bonds in there. And DNA wants to have, or at least in the beta form, is most stable when it has roughly 10.5 base pairs per turn. And so if you're underwound, then what you've done is essentially you kind of unwound a spring, and then what's going to happen is that spring's going to find a way to get back to a stable spot. And so what it can do is kind of form these kind of twists there. And so then it gets back from less than 10.5 base pairs to 10.5 base pairs. And same if you've overwound it, then what's going to happen is the DNA is going to twist as well. So instead of having 11 base pairs per turn, it's going to twist to try to get back to that 10.5 base pairs per turn. So if you watch those other videos, those short ones there, they do a really good job and have some better examples of this. So actually supercoiling is not just for circular chromosomes. In eukaryotes, the linear form of the DNA is very large, so it's almost like the ends are joined or they're fixed relative to each other. And so really the DNA just can't rotate freely. So you do get this coiling in linear DNA as well. And so here is an example of this idea of twist and writhe. And so essentially twist and writhe go with each other. So as I add more twists, the molecule can either writhe to reduce that twist, or you're just stuck with those additional twists. So again, since DNA is already a helice, as we start to add more twists to it, the molecule is going to writhe to try to reduce that change that you make. Either you're trying to fit more than 10.5 base pairs or less than 10.5 base pairs in that strand. And just understand that the DNA is going to writhe and form these kind of coiled coils. Okay, that is pretty painful, at least to me. It's this idea of topology. And if you want to make your head hurt in topology, a donut and a coffee mug are of the same topology. And so this idea of this supercoiling is actually in the realm of mathematics called topology. And again, they say that a donut and a coffee cup actually have the same topology. So if you can trust them, yeah, more power to you. Okay, so the key though is that in our bodies, DNA isn't just found in the linear form. It's often found in these twisted forms to increase the compactness of it. But in order to process the DNA, we're going to have to undo these twists and open up the DNA. And so you can imagine that if this is twisted, then you're going to have to untwist the DNA and it needs to respond. And so in doing so, you're going to add some of this strain, and the molecule is going to essentially do the supercoiling. Okay, so in that other posted video, they talk about topoisomerases. I'm going to kind of leave it to them. We really haven't talked about proteins, and so I won't expect you to know much about it because they're talking about protein mechanisms, and we really haven't even talked about amino acids. Just understand that if we have, like in this slide here, we have DNA, and we need to get some machinery in here to do something. And that do something we need to talk about. We either need to replicate the DNA or make an messenger RNA. We've got to be able to get in there. If we just took this molecule and tried to fit in here and expand that out, it's a little hard to see, but as I do that, imagine pulling these groups out here. And this really makes more sense actually if you just have two pieces of string that you twist around each other. And then if you start to pull out the middles, what you'll notice is that you're going to increase the twist here. Okay, or if you've ever just taken two pieces of string and just twisted them together, and you keep twisting them and keep twisting them, like I said with the rubber band, eventually you're going to see kind of those ends pull in, and you're actually going to get a shape that looks a lot like this. Oh shoot, my screen's not working. But anyway, I'll show you that in just 1 second. Okay, so like I was mentioning, if you twist up two strings, they'll eventually start to form shapes like the one on the left there that we see. Okay, and again, the goal for that is that we need to be able to access inside this duplex to do some interesting things that we'll talk about later. Now, the next little section of this book, again, this is chapter 24, so they assume that we've talked about proteins. They haven't. So when we talk about the covalent topoisomerases, just kind of forget about that and these structures here. It is interesting to be able to look at these structures, but we've got to understand what proteins are and even what these cartoon structures are. So sorry I didn't let you know earlier, but yeah, as far as the mechanisms and the interaction with proteins, we're going to skip over that. Now we can kind of process this diagram here and just understand how you would actually undo winding. And you do that by essentially nicking one of the DNAs. So if you are crossed over, you're going to have to nick a DNA, and then you're going to be able to pass one strand through the other. And in doing that, you can either wind or unwind the DNA. Okay. So, like I said, we're going to ignore these sections here. And just understand that a topoisomerase at this point just can wind and unwind the DNA. And let's get to something that we need to know about. Again, the whole point to this is if you start to unravel something that's twisted, so here, if I start to pull on these two strands here, and the other end is fixed here, so they can't really twist around each other. And you can do this with a piece of string. You start pulling those ends, you're going to compact the number of twists here. You're going to increase the twists in this area. So imagine that the twists here now have to fit in this area. And so you're over winding it. And so again, this happens naturally with just string. And what happens is they start to writhe and start to get these coils on top of coils. And again, these topoisomerases are what relieve this stress in the DNA when we need to process it. Okay, the whole point to all of that was I just wanted to get to this, where we could actually look at some organic molecules. So these topoisomerase inhibitors are actually chemotherapeutics. And so you may have heard of some of these drugs, so doxorubicin, ciprofloxacin, camptothecin. So these inhibit the ATP ACE activity or increase the rate of strand breakage. And so when you start to increase the rate of strand breakage, if you're doing this in an organism other than the human, that organism is going to die. They're not going to be able to seal up those nicks, and that eventually will lead to that organism's death. So some interesting drugs that stop this winding and unwinding of DNA. Okay, so in this section, and this is probably the most important section that we want to get to in 24, and that is the forces that stabilize the nucleic acids. So in DNA, we talk about the hydrogen bonding. And when I was young, I might have just been dumb, probably was, but at least it was taught back in high school that the number of hydrogen bonds is what stabilized the DNA structure. And that's not true. There is a little bit of stability, but if you think about it, DNA and those hydrogen bond base, nitrogenous bases can also hydrogen bond with water. And so to remove that DNA from hydrogen bond with water and base pair doesn't necessarily seem as energetically favorable as it once was. So there must be something else going on. So in the forces that stabilize the DNA, we want to look at other interactions that occur. Now, just as a side note, what I've shown here are actually some alternative ways that you can actually get base pairing, and we'll talk about that. What we've shown you earlier was actually the standard Watson-Crick base pairing, but if you start looking at these nitrogenous bases, there's actually lots of complementary patterns that can form between molecules, such as if I look here, adenine can actually base pair with adenine. It needs to twist around, but it does have complementary base pairing there. And in fact, these pyrimidines can base pair with pyrimidines. So now, if this happened in DNA, that would actually be kind of a constriction, because typically you have a purine with a pyrimidine. So this would actually be a little bit of a narrowing of the DNA. And our body does recognize when the DNA isn't as wide as it should and would come in and repair that. Okay, but DNA has a simpler job, but it needs to be very stable and convey genetic information from one generation to another. So RNA is actually much more complicated than DNA. RNAs are usually single-stranded and they fold up. RNAs actually need to be able to adopt a bunch of different shapes, and maybe not need to, but they've adapted to be able to take advantage of multiple different shapes and functions. And DNA also gets what we call post-transcriptionally modified. So actually our bodies do chemistry to the actual RNA strand. And so this picture is showing some of the different types of hydrogen bonding that we can get. And so if you see here, you get base pairing across the top of adenine here with thymine. And you can also get base pairing between thymine and adenine in this direction. So if we just compare these two, what it is it's a confirmation of the adenine. So here we've got an anti-anti, and here we've got a syn-anti. The take-home message from this is you can actually get a lot of different types of base pairing. So this is the typical GC base pairing, and then Okay, so this alternative type of base pairing is actually called Hookstein base pairing. And so these pairs off to the right are referred to as Hookstein base pairing. So there's alternative types of base pairing. And so, in fact, you can actually get triple stranded base pairing. And that can be involved in the regulation of DNA. So, like I said, you can even get triple base pairing. And so sometimes in DNA, you can actually get these types of stabilizing structures, but more often these are actually seen in RNA. So here is just a couple ways that you can get these triplex DNAs. So you don't really need to know these, just understand that there's more to DNA than simply the Watson-Crick base pairing. That those hydrogen bond patterns that are found on those nitrogenous bases can actually hydrogen bond with any donor and acceptor and lead to all sorts of interesting structures. In fact, one of these interesting structures is what's referred to as the G quadruplex structures, and these are found at telomeres. Now, we're not going to talk about telomeres in this class. In Biochem 2, you would, but these are found at the end of DNA. They're thought to be important in aging. Each time you replicate your DNA, your telomeres get a little bit shorter. So there used to be some thought that if you could find a ways to extend out your telomeres, you could live longer. I believe there is some truth to, well, not for humans, but there's some truth to keeping your telomeres, the ends of your DNA, in order is important for health. If you tend to lose your telomeres fast, it does lead to some diseases. Anyway, so at the end of your DNA, you have to find these G quadruplexes that kind of cap your DNA and prevent it from unraveling. And you get this G tetramer that forms. So you got these strands. So you got essentially a single strand that forms this tetramer and kind of blocks up your DNA. How I look at it is it's the end of your shoelace, that little plastic part that keeps your shoelace from unraveling. Again, you could talk about that later. If you take Biochem 2, they probably exist in telomeres, so the protective structures at the end of linear DNA molecules. And so it just happens that these four G bases, this G quartet, actually forms a very nice structure. Okay, but really what we want to talk about that stabilizes DNA is the base stacking. And so in base stacking, what you get is these heterocyclic bases that are overlaying another heterocyclic base. And there is some energetic interaction. Some people talk about these p orbitals interacting with these p orbitals, since these nitrogenous bases are aromatic. But there is some type of attraction between these bases. In fact, you can see what they've done here in this table to the right is they've calculated the stacking, or they determined the stacking energy for certain dimers. So if you have a GC and then a CG followed by a GC, you actually get the most stacking energy. For some reason, having this CGGC dimer is very energetically favorable, meaning that this C here is interacting very positively with that G, and this G here is interacting with that C beneath it. Some type of electrostatic interaction there. Now, it just happens that CG actually forms 3 hydrogen bonds. So, like I said, when I was young, we were taught that having those 3 hydrogen bonds, it took more energy to separate GC-rich regions. And it was easier to separate these AT regions, meaning pull and break those strands apart. Well, that's not the case. It's actually due to this base stacking. So you don't need to memorize this table, but you should understand that CG base stacking is much more energetically favorable than AT base stacking. Okay, and so again, it's the base stacking here, not the hydrogen bonding, but the interaction of these bases down this duplex that actually stabilizes the double helix. It's not the hydrogen bonding between the strands, it's the base stacking over each other. And so you get maximized base stacking when you get these two strands to come together, and then you get this base to interact with that one, and this base to interact with the one beneath it, and this one here. And so you get all this base stacking interaction. If you are just a single strand, you really don't get much base stacking other than what your sequence originally gives you. And you can imagine that not being in a helix, you're going to have a lot more mobility around that backbone there. And so DNA, you can actually separate these strands by heating it up. And it's what's referred to as melting the DNA or denaturing it. Now, it just happens that when you denature the DNA, it actually has a higher absorbance in the ultraviolet region. Around 260 is usually where you monitor it. So the denatured strand actually has a higher absorbance. So what you do is you put your DNA in there and then you start to heat it up. And what you'll see is the absorbance here increase until you get to a maximum, and then you know that you have separated or melted your DNA strands. Now, if we melt our DNA strands, do they ever get back together? Well, it depends. If you drop the temperature rapidly, then what you do is you just get these kind of random aggregations within the strand and between strands. And so DNA actually has this transition temperature. So if you look at the graph to the right here, we've got temperature and you've got the absorbance at 260. And so you can see that at a certain temperature, that absorbance is maximum, and that means that all the strands are separated, and here is the minimum. And so as we heat up, it melts, and those strands start to separate. So this midpoint is referred to as the TM. And so to get DNA back into its perfect helix, you don't want to get it super cold. So if you just get it and dunk it into an ice bath or even room temperature water, you'll just get random aggregation. What you want to do is just lower the temperature a little bit below, roughly 25 degrees below the melting temperature, and that has enough energy that these base pairs can break and reform and break and reform. And so if you leave it there for a while, you can get what they call annealing, or those base pairs to form perfectly again. So you don't want it cool enough for large paired sections to form. What you do is you want it cool enough that base pairs start to form, not too hot that it melts, but that this still has some random motion. And as it starts to make and break bonds, it will eventually adapt the most stable arrangement, which is with all the base pairs with their complementary pair in a double helix. Okay, so it's those base pairs that give rise, the base stacking that gives rise to the stability of DNA, but it's the base pairing that gives rise to the information and genetic ability to carry information in DNA. Now, like I mentioned earlier, RNA actually has a lot more complex structure. So it doesn't really form duplexes with itself. If you just have a single strand of DNA, it will start to fold on itself, but it tends not to form a duplex with another RNA. And so you actually will get where you get complementary sequences in there, and you start to get these loops. And then when you don't have complementary sequences, such as this region here, or this region here, or this region here, then you get these bulges or internal loops. And like I said, you can get base pairing between just the pyrimidines and other types of base pairing. And so here's an example of an RNA. And so it has a very complex structure. It's a single strand, but in here you've got the standard, what would call the canonical Watson-Crick base pairing, as well as alternative Hoogsteen-like base pairing. Again, the take-home message is that RNAs can actually adopt a lot more complex structures. And so here is the structure of a tRNA. So when we get into protein synthesis, we'll talk a lot more about tRNAs, but understand that they have this structure that is different than just a standard double helix. And so here is a interesting type of RNA molecule. It's actually called a ribozyme. So it's an RNA enzyme. These were first discovered in 1982. Now, RNA is actually involved in many processes in the body. In fact, it's involved in the synthesis of DNA at 3 different levels. And again, we'll save that when we talk about protein synthesis. But what's interesting about this ribozyme is it actually hydrolyzes itself. It sets up the bases, so you actually get a reaction within itself. So it's not really a catalyst, because the catalyst should be able to turn over and be reused. But this molecule does set up itself, so it actually does a reaction on itself. And that is known as a ribozyme. And here is just a picture of how those bases interact. And you can see that you get non-standard Watson-Crick base pairing in the active site there. So the mechanism for this is very similar to a base-catalyzed self-hydrolysis. So you're getting a hydrolysis of the RNA. And what I'm going to do is switch to why RNA is actually less stable. And that's due to this 2' hydroxy group right here. A 2' hydroxy group in the presence of a base can actually be deprotonated. And then when it's deprotonated, it can actually come over and attack the phosphate that's on the 3' position. And in attacking that phosphate, you do a substitution and you actually can kick out those electrons, and you've actually broken the strand here. So RNA, having that 2' hydroxy group, is actually less stable. And again, here's another diagram from your book. That 2' hydroxy group attacks the phosphate and breaks the strand. So RNA can actually be broken down fairly quickly by this mechanism. Enzymes can help, but even basic solutions will chew up your RNA. Now, I'll leave it to you to try to figure out if DNA has this problem. And that would be an excellent test question. Another issue with DNA that allows it to be genetic material is the presence of thymine. Now, this is cytosine right here. It's found in both RNA and DNA. Oh, DNA. And this is uracil right here. Now, cytosine in the presence of water will actually undergo what's referred to as a deamination. This is actually like an enamine right there. And so from organic chemistry, we know that enamines can be hydrolyzed and turned back into their keto form. So this is a little bit different than that. But same general idea is that you can hydrolyze this amine or deaminate it, and you convert it into a C double bond O. So another excellent test question is, what would be the problem if uracil was found in DNA? So the only difference in DNA is we have thymine and it has this methyl group right there. And that's actually very energetically costly to make thymine with this methyl group there. So why doesn't DNA just use uracil? And the answer is right here. So a good question on the test would be, what problems would arise if DNA used uracil instead of thymine? Okay, that ends up the forces that stabilize the nucleic acid structure. And we're quickly going to go into this fractionation of nucleic acids. And we'll talk a little bit more about this as we return to chapter 3. And so here, what we want to do is just be able to purify DNA. So when they talk about fractionation, you want to be able to isolate different fractions. And so one of the main ways of doing this is electrophoresis. If you've had genetics, you've had experience with this. I believe they talk about this in Gen Bio. Our book doesn't give a lot of details on it, but this is a very important technique. And essentially what you do is you have some matrix here. Now that matrix can be agarose or polyacrylamide gel, something that is networked, a polymer that's networked, but it has holes. So it's like a mesh. And what you're going to do is run a charge through there. And I'll give you a second to try to figure out what is the overall charge on nucleic acids. And you should be screaming the overall charge on nucleic acids is negative due to the phosphate backbone. So if I put this negatively charged molecule and I put a potential across there, or I run a current, this negative charge is going to run towards the anode or the positive side. So we can get our DNA to run from the negative side towards the positive side. So it's attracted to that. Now, if we have a network there, the molecule is going to be hindered in its travel. And you can imagine that larger DNA molecules are going to have a harder time traveling through there. And so as we run this in here, in this electrophoresis, what we're going to see is that we can separate out the DNA based on its size. And so the larger the size, the harder it is for it to run through this network there. And so what you're going to find is small pieces down here and large pieces up here. Okay, so here's actual example. Now, what they did was they just took DNA and they did a restriction endonuclease that we're going to talk about in just a minute. So every single strand or every single lane here, A through H, is the same DNA, but they mixed it with a different enzyme that cut it up into different size pieces. And so here we have fragments that range from 23,000 base pairs to just a little over 2,300 base pairs. Now over here in lane I is a standard marker. So you buy this from a biotech company and they've done a lot of work and you know the size of each band. So this band would be right. Ooh, actually, let's start at the bottom. It'd be easier. So that band goes there, and that band goes there. And this isn't matching up perfectly. Ooh, yeah, I'm missing some bands there. I might have pulled up the different marker, but essentially this marker here, sorry about that, you would know the weight of this because it comes from a company and they sell it as a marker. And then if I know this one here, then I could predict the size of the corresponding bands there. Actually, I'm thinking what happened was several of these bands are actually getting caught up in here. And so I'll tell you why that happens, and that is if your gel is too thick, meaning it is a very dense, that network there, you have very small holes. And so the DNA actually can't get through there. And so it gets caught up. So I'm guessing that's what's happening in this example there. But for now, just understand that in electrophoresis, we can separate these small fragments. But then how do we visualize them? Well, they take advantage of these dyes. Now, these are fluorescent dyes, so if you put them under a black light at the rave, they would look awesome. They have another property, and that is if you mix them with DNA, they intercalate, or intercalation. They slide perfectly in between those base pairs. So the stabilization, like we talked about, comes from this base stacking here, but there is enough room in there that another planar molecule can actually slide in there. And so these ethidium and proflavin and acridine orange, they actually just intercalate or slide in between those base pairs. And what happens when you do that is they fluoresce much brighter. And so if you run a gel and then you, let me skip that. If you run a gel and then soak it with ethidium bromide and then you soak it in water, what happens is the ethidium bromide or the dye washes away and it sticks to the DNA. And then when you put it under a fluorescent light, you can see where the DNA is actually found in there. Let me just go back for a second, just kind of show you what an agarose is. So you can set up an agarose gel and you can have different concentrations there. So like I talked about before, in that one gel, it was probably a little bit too dense. And so if you make it pretty dense, then you're going to be able to separate small fragments, but not necessarily large fragments. And there is another problem with this, and that is if you get really large strands of DNA, it becomes hard to separate because their migration is independent of size. And so what you do is do pulse-filled gel electrophoresis that the book talks about. We're going to see this when we return to chapter 3. So right now, understand that we can separate DNA based on its size in this electrophoresis. And there's other ways to separate DNA, but most of it takes advantage of its size or its charge. And so we talked about supercoiling in the DNA and how it changes its size. So here you can actually see that a supercoiled DNA, like I said, it should be more compact, and so it should be able to fit through these holes or this network in the gel much faster, and it does. And then if you nick it, you've started to unwind some of that, and here is just the normal dimer. So you can actually see if a molecule or DNA is supercoiled or not. Okay, there is another way to visualize the DNA on a gel, and that is to use what's referred to as southern blotting. So first of all, you'd run your gel electrophoresis, and then you want to visualize, and you don't want to use ethidium bromide. You want to maybe just isolate a specific band in there. And so you want to see if one of these bands matches a known sequence that you are interested in. And so what you do is you add NaOH, and that denatures and separates those double-stranded DNAs into single strands. And then what you do is you add nitrocellulose, also known as flash paper, but there is strong attraction between DNA and this nitrocellulose sheet. So you take the gel, you add some NaOH, and then you put this sheet of nitrocellulose paper, and then on the back side you put some paper towels. And what happens is they'll draw the water from the gel and start pulling that DNA, but the DNA is going to get stuck on the nitrocellulose. And then you've got this sheet with the DNA stuck to it. And then we want to visualize, like I said, a specific sequence. And so what you do is you have that sequence, but it has a radioactive phosphorus 32 label on there. And so that sequence, which is these little orange squigglies here, is only going to hybridize, only going to form a double helix with those strands that it's identical to. And then what you do is you wash everything away. And remember, these are single strands here until I add that probe. Now the probe only, in this example here, hydrogen bonded with these two sequences. And so we know that that probe that we put in, that we would know the sequence, corresponds to this band here. And so if we followed that band across, we know the sequence, or at least part of the sequence of this band now. And that is called Southern blotting, named after Edwin Southern. Chemists being funny, they came up with a Northern blot. It's like a Southern blot, but you do it with RNA. And then there's a Western blot, and that's for proteins, and you use antibody binding there. So since the guy was Southern, they thought they'd do a Northern blot and a Western blot, and I don't know, someone's probably even come up with an Eastern blot. Okay, so we can fractionate DNA and separate it based on its size, and we can identify that DNA either by using dyes or by using that Southern blot method. So let me just go back. So this is actually out of chapter 24. So they assume a lot of information there. The reason that we talked about chapter 24 is it goes more in depth into the double-stranded nature of DNA. But let's get back to our sequence here, where we're actually talking about nucleic acid function. What are nucleic acids good for? Well, nucleic acids are good for carrying genetic information. And this was actually known a long time before the structure of DNA was even known. And so one of the examples that they did was Frederick Griffith in 1928. He took a strand of pneumonia, and it was a deadly strand, a virulent strand, and he injected it into the mice and they died. Okay, well, there is a related one. In fact, they refer to them as the S colonies, or the smooth, and the R colonies for rough. And you give the R pneumonia to the mouse, and the mouse remains healthy. So they tried to figure out, well, what was it, the difference between these two? There's some morphological difference, but in the cells, they look different, but they knew that they were kind of related organisms. So what they did was they took the heat-killed S cells and injected them in the mouse, and the mouse lived. Okay, well, that makes sense. Heat kills that virulent strain. Well, then what they did is they took the living R cells that, notice, didn't kill the mice, and they put the heat-killed S strain in there. And now all of a sudden, the mice died. So there was something in the S cells that didn't kill the mice by themselves, but in the presence of a functional cell could kill the mice. So there was something that transformed those non-virulent strains into a killer strain. And that was known back in 1928. And so here we have the a picture of the different ones. Now, the DNA that they used was highly purified, so it wasn't affected by enzymes that cut up proteins. It wasn't affected by RNases that cut up RNA. So they knew that it was DNA. And in fact, if they used an enzyme that ate up DNA, this experiment didn't work. So something, when they killed these cells, was still floating around in solution and could be uptaken by the non-pathogenic organisms and turn this into a killer. Okay, so that was 1928, and it took them many decades to actually get out the structure of DNA. Like I said, it's really interesting. They really didn't believe his results for a long time. They were eventually later replicated, and they started to believe that there was some transforming material. Like I said, they thought it was proteins for a long time, but it was evident that it was nucleic acids. And so once they got the structure of DNA, they realized how the genetic information was transferred from one generation to another. And so what we have here is a double-stranded DNA. Each one of these strands is listed as old. Now, each one contains the complement to the other one. And so if I want to pass on information to someone, if I just had a letter and I gave you that letter, I no longer have a copy of that information. If I need to retain that information for my own knowledge, what I would do is make a copy of that letter and give it to you. So back in the day, monks would take books and they would make copies. And when they made those copies, sometimes there were errors, but essentially monks just sat around and made copies of books and then sent them around until you got Gutenberg and the printing press. Well, you can look at DNA as kind of like a printing press. It's got this copy of information. And so what you do is you separate the strands and then you just make the complement to each strand. And as you make the complement to each strand, eventually you're going to have 2 full copies. So that is how DNA is replicated. And again, we'll talk about that process a lot more, but understand that it's due to this duplex nature that DNA can serve as genetic information. We can use these complementary strands, copy each complementary strand, and then I have 2 copies of that information. And then if I took those 2 strands and copied them, I would actually have 4 copies. And if I took those 4 copies, then I would then have 8 copies. And that's going to be an important technique that we'll talk about, where we can actually amplify this DNA, because each time I replicate it, I'm going to double those copies that I have. Now, DNA, like I said earlier, doesn't do much. RNA actually is involved in many biological processes, but DNA really is just an information carrier. It doesn't carry out reactions. It's just like the hard drive in your computer. If we want things to get done, we need to make RNA, and going from DNA to RNA is referred to as transcription. I'm transcribing from essentially one language to, well, sorry, I'm transcribing, meaning copying from one to another, not a language. That comes in when we translate. So once we transcribe and I make RNA, which is just a different nucleic acid, again, it has that 2' hydroxy group, and it also has uracil in it. But what I can do is I can actually use tRNAs, which are RNA molecules that are actually covalently bound to an amino acid there. So notice here that I'm taking mRNA, so an RNA molecule, I'm taking another RNA molecule, and guess where all this happens? In the ribosome. And the ribosome has a large component of RNA in it. So it's really interesting that RNA is involved in carrying the information from DNA, bringing in the amino acids, and essentially providing the site and the, I guess, the chemical reactivity to make proteins. So we can go from DNA to proteins, but it requires this mRNA on many different levels. Now, your book has a little section on the RNA world. So to kind of bring this full circle, if we try to answer the question of, well, how did life arise? Well, we needed an information carrier. And we also needed molecules that can do chemical reactions. Well, we just talked about the ribozyme, and now we see here the messenger RNA is carrying genetic information. It's also carrying proteins. And so ribozymes are small molecules that can do chemical reactions. And then we got the ribosome, that's actually a large molecule that actually can make proteins. So it was thought, there's a hypothesis out there, that the first life molecule or genetic information molecule or life really started with RNA. And there is some hints that that could be true. Like I said, RNA is found in the stages of transferring information and involved in chemical reactions. Okay, so we can make proteins. Now again, we really haven't talked about proteins. So we're going to come back to this protein synthesis again. just understand that you go from DNA to RNA to proteins. Now, this is referred to as the central dogma. So it was developed by Crick to explain the possible direction of information flow in cells. So you could think that, well, DNA goes to DNA, RNA makes RNA, and proteins make proteins, or all possible DNA makes RNA, and all those are the possible routes. Well, what they found is that not all those routes are followed biologically. So Watson and Crick said, well, DNA actually can make DNA, and we just talked about that by virtue of having this double helix. DNA can serve as a template to make RNA, and then RNA actually is used, we read the RNA to make proteins. So this was the central dogma. Now, we found out that actually you can go from RNA back to DNA. This wasn't known as at the time, but that is a possibility. In fact, our friend coronavirus takes advantage of this, and it's an RNA virus, I believe. Someone email me if I'm wrong, but it's an RNA virus that actually goes into the cell and first makes DNA, and then that DNA is used to make copies of that virus. So we do know that you can go from RNA to DNA. So, but the central dogma of molecular biology deals with the detailed residue by residue of information. How do we transfer information from one of these units to the other? And so this is the central dogma that you may have learned, that DNA goes to DNA, but DNA can also go to RNA, and RNA can go to proteins. But like I said, we can do the reverse transcription here. And the enzyme that does that is reverse transcriptase, and it can make DNA again. So that is the central dogma of biology, or molecular biology, or really how information is transferred from parents to children or offspring. So most DNA molecules in a cell are huge. So the total weight of the human genome, we have roughly 3.3 times 10 to the 9th, should be an exponent, base pairs, roughly 650 Daltons per base pair. And Daltons is just the way that they measure. The equivalent would be grams per mole in chemistry. Oh, and that's 2.5 times 10 to the 12th Daltons. I'll fix that in the slides, my exponents, which is roughly about 3.6 times 10 to the negative 12th grams of DNA in a cell. And you have roughly 3.7 times 10 to the 13 cells, so that's roughly 130 grams of DNA in you. And if you go look at something 130 grams, that's actually quite a bit. And that doesn't include your microbes. In fact, there's more microbe cells in your body than there are you cells. So inside of you, there's more microbes, cellular-wise, per cell count than there are you. So if all of the chromosomes in your cells were unwound and placed end to end, your genome would be about 2 meters long. And so the E. coli genome is actually a little bit, quite a bit smaller, so only about 3 millimeters long. But a E. coli is only 1 micrometer in diameter. So let's kind of bring this all back together. So why did we talk about coiling and base stacking and all these other interactions? Well, this is a poor virus right here that they lysed it. And when they lysed that cell, it spread its DNA all over. And so you can see how much DNA was actually packed into this little virus right there. And so to bring this all back together, we've got to find ways to pack this very, very long molecule that has these base pairs that encodes genetic information that we eventually turn into proteins and actually into mRNAs and other uses in the cell. We've got to find a way to pack that. And so we pack that in helices that supercoil, that are stabilized by base stacking. The fidelity of the information is maintained by base pairing. And again, that is what allows us to exist, that genetic information found in the nucleic acids. So tomorrow we'll finish up this chapter. We'll talk about DNA technology and some of the things that you can do to DNA, how we know the sequence of DNA, how we can make transgenic organisms, and stay tuned for that.